Hello everyone, welcome back to the NPTEL session of overview and integration of cellular metabolism. So, we were discussing in our last two classes lipoprotein metabolism. This class is about lipoprotein metabolism once again. This is the last class of lipoprotein metabolism. So, to recapitulate the previous classes that we have discussed, the general structure, biochemical properties of different lipoproteins, then the metabolism of different lipoproteins like chylomicron, VLDL, LDL, HDL. Now, today we are going to discuss two important topics. One is reverse cholesterol transport, another is disorders related to lipoprotein metabolism. So, reverse cholesterol transport is important with regards to HDL function or high density lipoprotein function. So, if you remember the metabolism of HDL, that the nascent HDL, say it intestinal HDL or nascent hepatic HDL, they are initially secreted as pre-beta HDL, and this pre-beta HDL takes cholesterol from extrahepatic tissues. Then it forms the discoidal nascent HDL. Now, this discoidal nascent HDL, if you remember, is forming spherical matured HDL3. How? By uptaking cholesterol and cholesterol esters from the peripheral tissue. So, this part is known as reverse cholesterol transport. Why? Now, remember cholesterol, endogenous cholesterol in very low amount or exogenous cholesterol which is ingested through diet, that is very low in amount, and also endogenously, mainly endogenously synthesized cholesterol, they are delivered to extrahepatic tissues with the help of the lipoprotein particles like VLDL, most important is LDL, low density lipoprotein. So, this is how cholesterol is transported from liver to extrahepatic tissues. Liver is the synthesizing organ of cholesterol. So, this is cholesterol transport. The reverse one is basically transporting or transferring back of cholesterol from extrahepatic tissue to liver. So, this is known as reverse cholesterol transport, and this reverse cholesterol transport is done by HDL. Now, in this regard, there are two very important receptors that is playing those are playing the main role in reverse cholesterol transport. So, one is SRB1. If you remember, we have named these two receptors in our previous class. SRB1 stands for scavenger receptor B1, and ABC transporter protein. ABC stands for ATP binding cassette transporter proteins. So, these two groups of receptors are very important in reverse cholesterol transport. Now, SRB1 plays dual role. If you remember once again, you can see in the previous slide that as we have discussed that HDL2 is basically uptaken with the help of the receptor SRB1 in liver. So, through SRB1 receptor, the hepatic SRB1 receptor, HDL is basically transferring the cholesterol, or basically there is transfer of cholesterol from HDL to liver. So, SRB1 is playing the role of taking cholesterol from HDL inside the liver, that is one role. Another role of SRB1 is basically delivering of cholesterol from extrahepatic tissue to HDL. So, in extrahepatic tissue, the role of SRB1 is basically transferring the intracellular cholesterol to HDL. So, remember SRB1 receptor or transporter, it plays dual role. HDL uptakes peripheral cholesterol through SRB1 as well as it delivers its own cholesterol to liver through SRB1 transporter. Now, the other one, ABC transporter protein or ATP binding cassette transporter protein, it is basically one ATP dependent transporter. So, there is one ATP attached on binding with the ligand, these transporters, the bound ATP is hydrolyzed, the energy is the reason by which there is delivery of intracellular cholesterol from the cell to the HDL. So, ABC transporter protein. Now, ABC transporter protein are of different types. It can be ABC A1, can be ABC G1. Now, remember ABC A1 is important for delivering of cholesterol to the pre-beta HDL. So, once again, if we go to the previous slide, so you can see that ABCA1 is important for delivering cholesterol to the pre-beta HDL, whereas ABCG1 transporter is important for delivering cholesterol to the discoidal HDL as well as to the matured HDL3. So, this is important for reverse cholesterol transport. So, you can see pre-beta HDL is the cholesterol poor triacylglycerol depleted particle. So, it has the maximum tendency of uptake of cholesterol from the endogenous extrahepatic tissues. Then HDL3 are also, HDL3 molecules are also important for uptake of peripheral cholesterol. So, these two important receptors are there which are actually important for reverse cholesterol transport. Apart from that another enzyme, that is LCAT, if you remember, lecithin cholesterol acyl transferase is also important. So, basically the transferred cholesterol, cholesterol which is transferred from extrahepatic tissue to the HDL is actually converted to cholesterol ester with the help of the enzyme LCAT, forming the hydrophobic core. So, basically the content of cholesterol is reduced in HDL. Similarly, another protein is there, that is CETP. So, CETP, what it is causing? CETP is delivering the cholesterol ester from HDL to the other lipoproteins like VLD and LDL. So, basically these two, LCAT as well as CETP, they are reducing the cholesterol content in HDL. So, cholesterol depleted HDL formed when it circulates through the extrahepatic tissues. The gradients difference of cholesterol between HDL and extrahepatic tissue is the main reason of cholesterol transfer from extrahepatic tissue to HDL. So, basically whenever there is cholesterol poor HDL is circulating through the extrahepatic tissues which contains high amount of cholesterol, there is reverse cholesterol transport through the receptor SRB1 as well as ABC transporter protein. So, now we are going to discuss different clinical conditions related to lipoprotein metabolism. They are mostly related to lipoprotein synthesis, lipoprotein transport or lipoprotein degradation. Remember, these are the primary defects in lipoprotein metabolism. So, basically these are the inherited condition. Similarly, different other acquired conditions are there where lipoprotein contents are increased or decreased, importantly diabetes mellitus, atherosclerosis, kidney disorder like nephrotic syndrome. Those are the acquired condition where lipoprotein concentration is increased in the circulation, but today we are going to discuss the familial condition or inherited conditions, these are known as the primary dyslipoproteinemias. So, these lipoproteinemia can be hypolipoproteinemia, can be hyperlipoproteinemia. So, hypolipoproteinemia are basically where the lipoprotein concentrations are low, hypo, whereas hyper stands for increased. So, hyperlipoproteinemias are basically where the lipoprotein concentrations are increased. So, these are the two important primary hypolipoproteinemias. One is A beta lipoproteinemia. So, A beta lipoproteinemia is basically associated with a defect in synthesis of ApoB. Beta lipoprotein is related to ApoB, apoprotein B. Now, the problem in synthesis of ApoB, and if you remember, ApoB 100 is linked with LDL and ApoB 48 is linked with VLDL. So, what will happen if there is ApoB 48 is basically related to chylomicron, I am sorry, VLDL and LDL, they are linked with ApoB 100. So, what will happen? There will be reduced concentration of chylomicron, VLDL and LDL, because there is no apoproteins, no apoprotein B is there. So, what will happen? In blood, acylglycerol concentrations will be very low. So, there will be accumulation of those lipids or acylglycerols to those organs from where chylomicron and VLDL were synthesized. So, chylomicron were synthesized in intestine and VLDL were synthesized in liver. So, those triacylglycerol, because they cannot come in the circulation through chylomicron or VLDL, so they will be accumulated inside intestine and liver. So, there will be intestinal malabsorption. Those lipids cannot be absorbed and cannot enter the circulation. So, there will be intestinal malabsorption. And if you remember that dietary lipid is important for transfer of lipid soluble vitamins like vitamin A, D, E, K. So, there will be severe these fat soluble vitamin deficiencies, and the early death can be avoided by administration of those fat soluble vitamins in large doses, particularly vitamin E in this regard is very important. So, this is about beta lipoprotein deficiency. Then we are coming to the alpha lipoprotein deficiency. Alpha lipoprotein is related to HDL. So, there are three primary alpha lipoprotein deficiency. One is Tanger's disease, then there is fish eye disease, and finally ApoA1 deficiency. Now, what happens in Tanger's disease? This transporter synthesis is defective. So, if you remember ABCA1 transporter, why it was important? Remember pre-beta HDL, the nascent, very nascent HDL which is secreted from intestine or liver, it was pre-beta HDL, and pre-beta HDL takes up the peripheral or extrahepatic cholesterol with the help of the transporter ABCA1 to form the nascent discoidal shaped HDL, which is finally forming the other forms of HDL, that is HDL3 or HDL2. Now, in absence of this ABCA1 transporter, what will happen? HDL will remain in the form of pre-beta HDL. So, there will be no discoidal HDL, no matured HDL, and those were important for, if you remember, those were important for reverse cholesterol transport, those were important for different lipid exchange in between. So, those will not happen. Similarly, fish eye disease is another alpha lipoprotein deficiency where there is partial, remember there is partial LCAT deficiency. So, lecithin cholesterol acyl transferase enzyme partially deficient in fish eye disease. So, this LCAT is once again that is a lesson from the previous classes, that LCAT is responsible for converting cholesterol to cholesterol ester in HDL. So, that is forming once again the whatever cholesterol is actually uptaken from the extrahepatic tissues, those are converted to cholesterol ester and forming the hydrophobic core. So, those are the spherical. So, LCAT is responsible for forming the spherical HDL, mature HDL, that is HDL3, HDL2, those were formed by LCAT. So, once again the matured HDL will be absent in circulation. Similarly, ApoA1 deficiency is another important thing. If ApoA1 is deficient, there will be defect in pre-beta HDL formation even. So, finally, what will happen? There will be low or near absence of HDL in circulation. Now, if there is absence of HDL, remember HDL was acting as the store of different apoproteins. One very important apoprotein is ApoC2. So, if you remember once again that the nascent chylomicron and the nascent VLDL, they have been matured with transfer of ApoC2, and that transfer occurs from HDL to the nascent chylomicron or nascent VLDL, and that ApoC2 was important. Why? For activation of lipoprotein lipase. So, lipoprotein lipase was that important enzyme which is finally degrading triacylglycerol and providing the free fatty acid or free fatty acid to the peripheral tissues. So, that will not happen. So, lipoprotein lipase will not be activated because there will be no HDL. So, there will be no ApoC2 delivery and there will be no lipoprotein lipase activation. So, what will happen? The circulatory triacylglycerol will be increased because the matured chylomicron or matured VLDL, they will be having triacylglycerol inside them, but they cannot be able to deliver the lipids to the peripheral tissue. So, what will happen? There will be predisposition of atherosclerosis because of increased circulatory triacylglycerol due to absence of HDL. So, these are the two hypolipoproteinemias. Next, we are coming to hyperlipoproteinemia. So, primary or familial hyperlipoproteinemia are of different type. Amongst them, type 1 is due to lipoprotein lipase deficiency. So, type 1 is known as familial lipoprotein lipase deficiency, where the function of lipoprotein lipase is defective. Now, this defective lipoprotein lipase can be due to deficiency of lipoprotein lipase or the factor which is activating lipoprotein lipase, that is ApoC2 deficiency, that is also causing inefficient lipoprotein lipase function. So, finally, the effect will be there is no delivery of triacylglycerol or no breakdown of circulatory triacylglycerol in chylomicron and VLDL. So, the clearance of those chylomicron and circulating VLDL and chylomicron will be low, but it happens. So, coronary disease risk is comparatively low in this condition. Now, coming to the type 2A, which is known as hypercholesterolemia, and this is due to the defect in LDL receptor. Now, remember LDL once again, where we have discussed the structure of LDL receptor. Like all receptor, there is a ligand binding domain. Now, in type 2A dyslipoproteinemia, that ligand binding domain of LDL receptor is basically defective. So, even if there is LDL receptor, they cannot bind the ApoB100. So, what will happen? LDL will not be catabolized or uptaken inside the liver or inside the peripheral tissues. So, there will be elevated LDL in circulation causing hypercholesterolemia. Circulating cholesterol is very high and that will finally be deposited, those cholesterol will be deposited in coronary vessels, heart vessels, causing atherosclerosis. Next condition is type 3. So, type 3 hyperlipoproteinemia is also known in different other names like broad beta disease, remnant removal disease, familial dysbeta lipoproteinemia. Now, this remnant removal disease, if you remember, you can remember what happens in this type 3 condition. What happens is basically there is a defective remnant removal. Remnant means chylomicron remnant or VLDL remnant as well. So, what will actually, where is the defect? The defect is in ApoE. Now, remember the remnants, VLDL remnant, IDL or chylomicron remnant, they were uptaken by the receptors and those receptors were actually sensing ApoE in those remnants. So, if ApoE is defective, in that case chylomicron remnant or VLDL remnant, they will not be uptaken inside the liver. So, there will be definitely more circulating chylomicron and VLDL remnant that will be causing hypercholesterolemia, deposition of those cholesterol in skins causing xanthoma as well as atherosclerosis. Next is type 4 familial hypertriglyceridemia. Here is overproduction of VLDL. VLDL is produced in huge amount and this condition is associated with glucose intolerance as well as hyperinsulinemia. So, basically this type 4 condition is associated with different other metabolic condition like coronary artery disease, coronary heart disease, type 2 diabetes mellitus, obesity, alcoholism, and it is also induced by progesterone based hormones, administration of progesterone based hormones. So, this is about type 4 hypertriacylglycerolemia. Then familial hyperalpha lipoproteinemia, HDL concentration is increased. So, this is a sort of beneficial condition. Then there is deficiency of hepatic lipase. So, that hepatic lipase was important for clearing the residual triacylglycerol present in the remnants of HDL or chylomicron, those will not be cleared. Then familial LCAT deficiency, lecithin cholesterol acyl transferase enzyme deficiency, which is important for reverse cholesterol transport. So, if there is LCAT deficiency, the reverse cholesterol transport will be hampered. Then another important one is familial lipoprotein A excess. Now, lipoprotein A is a specialized form of apoprotein which has this lipoprotein has ApoA. Remember this ApoA, A is in small letter. Now, this ApoA has the structural resemblance with the plasminogen. Now, this plasminogen is basically inhibits the clot removal. So, this is a proatherogenic form, causes atherosclerosis because it inhibits the clot removal, it inhibits the clot resolution. So, with this type of this familial lipoprotein A excess condition of dyslipoproteinemia, definitely there is predisposition of coronary heart disease, premature coronary heart disease due to atherosclerosis, and as well as different other vessels are more predisposed to thrombotic blockage because there is inhibition of fibrinolysis because of this ApoA. Plasminogen is important for fibrinolysis, remember. Plasminogen is important for this clot removal or fibrinolysis. So, that clot removal is inhibited because of ApoA, so causing thrombosis in different other vessels. Now, coming to the important role of LDL in causing atherosclerosis. Atherosclerosis is basically narrowing of vessels, narrowing of circulatory vessels due to deposition of different forms of lipid as well as different inflammatory cells. So, finally, there is low circulation, low blood circulation through the vessels due to narrowing of the vessels. Now, LDL plays one very important role in this atherosclerosis pathogenesis. Now, atherosclerosis is a broad, the pathogenesis of atherosclerosis are having different hypotheses, different phenomena. We are highlighting how LDL is contributing to atherosclerosis. Now, remember LDL while going through the circulation, if it remains in circulation for more time, it is predisposed to different cellular environment. Now, where oxidative stress or free radical generation is high, in those conditions LDL or low density lipoprotein can be modified to different forms. One very important form is ox-LDL, that is oxidized LDL. Now, this oxidized LDL is important with regard to pathogenesis of atherosclerosis. Now, what happens when there is more oxidized LDL formation? The receptors which uptakes this oxidized LDL are synthesized in more quantity or more amount. One such receptor is LOX1. Now, LOX1 is known as lectin-like oxidized LDL receptor 1. Now, this LOX1 receptor is highly expressed in endothelial surface. Endothelial means blood vessels, endothelium. In this blood vessels endothelium, LOX1 receptor is expressed in high amount when there is high circulation of oxidized LDL. Now, LOX1 binds to the ox-LDL, that is a receptor ligand binding, and it delivers the oxidized LDL inside the intima media of blood vessel. Now, in this intima media there is a pro-oxidant environment, there is already oxidative stress in the cellular environment which has already triggered the inflammation. Inflammation has caused intrusion of macrophages inside the intima media. Now, these macrophages are very much predisposed to uptake oxidized LDL. Now, the oxidized LDL which is bound to the LOX1 receptor is uptaken by macrophage by various type of scavenger receptors like scavenger receptor A1, remember this is A1 not B1, then CD36, LOX1, these are the scavenger receptors which takes oxidized LDL from the endothelium. Inside the macrophage, this oxidized LDL is processed. How processed? Definitely it is uptaken by receptor mediated endocytosis. That endosome formed is fused with the lysosome, forming endolysosome. In the lysosome there is lysosomal acid lipase. Lysosomal acid lipase degrades the cholesterol esters of oxidized LDL to free cholesterol and fatty acids. Now, this free cholesterol once again can be esterified by another enzyme known as ACAT1. ACAT1 stands for acyl coenzyme A, acyl transferase. Now, this ACAT1 basically re-esterifies the free cholesterol inside endoplasmic reticulum. Now, this stored cholesterol in cholesterol ester in endoplasmic reticulum can be degraded with the help of the enzyme neutral cholesterol ester hydrolase to release cholesterol, free cholesterol, and that free cholesterol can be uptaken by HDL, if you remember, by the reverse cholesterol transport. Now, problem is when there is a pro-oxidant environment in cell which is predisposing to which is causing release of different cytokines, different pro-inflammatory cytokines, accumulating different pro-inflammatory cells, pro-inflammatory macrophages. In those conditions, what happens? These receptors related to reverse cholesterol transport like ABCA1 receptor, ABCG1 receptor, SRB1 receptor, these receptors are down regulated. So, the reverse cholesterol transport is hampered. Remember, when there is a balanced condition, when there is a homeostasis in lipoprotein metabolism, even if this oxidized LDL is entering in macrophages, they can be delivered to liver by a reverse cholesterol transport. But the problem happens when there is the pro-oxidant condition or inflammatory condition is exaggerated, so much so that the homeostasis cannot be maintained. So, in those cases, these receptors related to reverse cholesterol transport are very much down regulated. So, the reverse cholesterol transport is hampered, as well as this ACAT activity is very high. The macrophage receptor which are causing uptake of oxidized LDL, they are expressed in very high concentration. So, what happens? There is increased uptake of oxidized LDL in macrophages. Those LDL are basically delivering cholesterol, and the cholesterol esters are accumulated inside macrophages which cannot be cleared by reverse cholesterol transport. So, the macrophages are now highly full with the cholesterol ester, forming foam cell. Now, those foam cells are actually accumulated in intima media of the vessels, causing formation of fatty streaks. You can see there is formation of fatty streaks, which is, you can see, causing narrowing of the lumen. So, the lumen diameter is narrowed down, and finally, they are forming atherosclerotic plaque. Now, this atherosclerotic plaque is important for narrowing of blood vessels, finally predisposing to myocardial infarction. So, this is how LDL contributes to formation of atherosclerotic plaque, finally causing atherosclerosis and myocardial infarction. So, another important role here I want to discuss is that the role of CETP. Remember, CETP was important for exchange of triacylglycerol and different cholesterol esters in between HDL and other lipoproteins like VLDL or IDL or LDL. Now, the problem is when there is inhibition of CETP, the HDL3 cannot be converted to HDL2. Remember that there is a cycle of HDL. If you remember that HDL3 was actually forming HDL2 by donating by uptaking triacylglycerol from VLDL or IDL with the help of the protein CETP. Now, this cycle will be hampered, and remember HDL2 was actually used to be uptaken by liver. So, what will happen? There will be increased concentration of HDL3 in absence of CETP. This conversion to HDL2 will not happen. Similarly, HDL2 will not be uptaken by liver, because that is not HDL2, that is HDL3. hepatic lipase is acting less. Also, the scavenging function of HDL will be low. So, finally, CETP inhibition is associated with atherosclerosis. Now, there are different drugs which causes inhibition of CETP, those are actually predisposing to atherosclerosis. So, what we have seen that LDL is related to positively related to the risk of cardiovascular disease, but the role of HDL is not absolute. Remember, even if HDL3 is very high in circulation, that is causing atherosclerosis. So, what is required is LDL HDL ratio, which can be used as a diagnostic tool for cardiovascular disease. So, the good one, LDL HDL ratio is taken as 3.5. So, in this session what we have learnt is a detailed reverse cholesterol transport, how HDL is actually transferring back the peripheral or extrahepatic cholesterol to liver. Then the role of HDL in atherosclerosis via formation of foam cells through different modified LDL like oxidized LDL, and also we have discussed different primary dyslipoproteinemia or primary lipoprotein metabolism disorder. So, these are my references. Thank you all. See you in the next session.